Hello and welcome to my next and final AS live stream on Unit 2 Biology. Just checking, is everyone ready and is all the audio and visual stuff working fine? Okay, great. Right, so first question. January 2013, Unit 2, question 5D. Yeah, it's a ridiculously annoying question. Right. Two examples of such international corporations are CITES, and the Rio Convention. Other than the conservation of biodiversity aim, right? Well, this is in, I think, like literally the final page of the whole biology book, is it? Yeah, it is the final page, and it talks about all these random stuff. Basically, CITES is to regulate and monitor, prevent trade in endangered species, ensure trade does not endanger species, prohibit trade in wild plants, allow trade in artificially propagated plants, allow trade in less endangered species. It's just to do with trades, basically. And the Rio Convention is sustainable use of organisms, habitats, and ecosystems, share genetic resources, share access to scientific knowledge, technology, and knowledge, promote ex situ conservation methods. So that's like zoos and seed banks. Raising profile of biodiversity with governments, public bodies, and the general public, and international cooperation. What was your question before, James? Describe the structure and mode of action of T and B lymphocyte. For person who asked transmission, no, sorry, not Mark. When you said structure and mode of action of T lymphocytes and B, is that OCR? Because I don't think that's in the OCR specification. If it is, I've never seen it. So could you just clarify that, please? The causes and means of transmission of malaria and AIDS, HIV. Yep, sure. Right. Malaria is caused by the Protoctist Plasmodium, I believe. Yep, Plasmodium, good. And it is carried by the female Anopheles mosquito. And basically, the female Anopheles mosquito sucks blood, and then the Plasmodium, it sucks up the blood with Plasmodium in it. Then, once it's got that, it'll bite an uninfected person, because the Plasmodium will have where was I? Sorry. So the Plasmodium develops and migrates to mosquitoes' salivary glands, which then bites another person, injects the Plasmodium. The Plasmodium migrates to the liver and migrates to the blood, replicates, and then you're infected with malaria. That's malaria. HIV is caused by the human immunodeficiency virus, which after a while becomes AIDS, which is autoimmunodeficiency syndrome, I believe. If I am wrong with that, please correct me. I believe it's autoimmunodeficiency syndrome. And that is transferred by basically interaction of bodily fluids. So blood-to-blood contact, unprotected sexual intercourse, unscreened blood transfusions, sharing needles, anything like that which shares bodily fluids. Right. Microtubules move all. I can't really draw it, but actually I can. Right. Right. Cell, nucleus. Now there are kind of these are you have microfilaments, I believe. This is the cytoskeleton. So you kind of have highways which kind of take things all over the cell. So you might have something that goes to the nucleus. You might have something which goes to a lysosome there, or something that goes to Golgi. I'm not going to draw it, but Golgi. And then what happens? Microtubules are things that can kind of rub together. So let's say you have mitochondria. It might have microtubules on it. These microtubules rub together with friction, will cause movement and causes the mitochondria perhaps to move along the cytoskeleton. There you go. That's basically what it is. I hope that explains it. I don't think you need to know that. Oh. Even Alice in Gymnast, I would still like confirmation. Just what? So I have a feeling Mark, if I remember, if I'm remembering, I'm sorry, but I believe he doesn't do OCR. I think he might do another exampled, which is why. No, we're doing Unit 2 now. We did Unit 1 earlier. Um, what do you exact same book size do for the for biology? Um, Structure of antibodies. Right. Right. Yeah. Just I will I'm answering a question now. Any questions that are asked, I will get to. I just have to do them in order which they are asked. Right. So, right. This is the structure of an antibody, and then you have. And you have this chain here is called the heavy polypeptide chain. It's the long one. The smaller one is called the light polypeptide chain. These are disulfide bridges. Right. You then have the constant region, because it's always the same for all antibodies. Because this constant region means it can bind to any normal cells and be recognized by the body as its own body. One sec. Page 170. Okay, obviously, marks. Oh. So when he said describe the structure, I thought he wanted in a lot more detail than I knew about. Obviously, okay, right. I will answer that next. So I thought he wanted like full structure like you have with antibodies. Sorry about that. Right, sorry. Right. Anyway, so yes, revision pays off. Right, constant region. This is the variable region. Is different. So that's going to be complementary to a specific antigen. I believe that's the full. Is that everything? Oh yeah, and finally, here is the hinge region. So heavy chain, light chain, disulfide bridge, hinge region, this bit here, constant region, variable region. Right, so that's the structure. And then, right, you have neutralization and agglutination. Right. Neutralization is you have pathogen, and it'll be, I'm going to draw a Y for an antibody. It's going to be neutralized by many antibodies. So antibodies cover the pathogen. And the other one is the opposite. You will have an antibody which could have many Y sections, which will then cause a pathogen to bind to it. So that's agglutination, is when the pathogens bind to an antibody with many variable regions. Neutralization is when you have many antibodies binding to one pathogen. Which the heavy one is the long one. Look, heavy, long, light, short. Heavy, long, light, short. Yes, it is in the immune system video. Right. So okay, right. Well, hope that answers your question. Whoever asked. No, it's just the long one is the heavy one. The light one is the short one. Right. Okay. Well, the structure of B lymphocytes and T, they all have receptors which are complementary to the shape of the foreign antigen. T lymphocytes have receptors. B lymphocytes actually have the antibodies on them, so that's their sort of receptors. Right. So that's basically the structure of them. And I believe they are proteins. They are probably proteins. I'm not 100% sure though. Actually, no, they're cells, aren't they? Sorry, they are cells. The receptors, that's important. The receptors are the proteins. That's what I meant. Right. Right. And these cells can release cytokines, which can communicate with each other to kind of go, look, this is the problem we've got. What's the purpose of that? It's just there to kind of make the shape. If you since you have the heavy and the light, you can kind of have that kink in it. And the heavy and they're just part of the main structure. Can we go through the whole immune response? Yeah, sure. Right. Well, you have this communication between the B and the T lymphocytes. That's the cell signaling. So they release these cytokines, which will then communicate with other cells, and to say, look, this disease is here. Let's make more of ourselves to destroy it. In terms of memory cells are there, so if you get the disease again, your body will be able to go, okay, right, we know what this disease is now. Let's quickly get rid of it, and you should not suffer any symptoms, and it'll be dealt with much quicker. So I hope that answers your question, Mark. Please tell me if it doesn't. Right. Aubrey, no. So I have videos on everything on AS biology. Every video has been covered and every topic has been done. So any topics you don't understand, please feel free to. I will answer them for you. Yeah, I will answer the immune. The immune system isn't actually that long. I have done it in a previous live stream though. So have I got any other questions to answer before I go on to that one? Let's just see. Okay, I'm just going to explain the role of DNA and RNA in protein synthesis. Okay, right, DNA nucleus, you have DNA. double-stranded. DNA has the genetic code which will code for the protein. And then what happens during transcription, mRNA, so zoom in over here, you'll get one strand of DNA like this, and mRNA will bind to it. It'll be complementary binding, except the T base is replaced with U. So we have here A, C, G, T. You'll have U, G, C, A, because it's complementary. And this mRNA then leaves the nucleus and goes to a ribosome where it will then translate and be coded for, and then tRNA will bring amino acids for each triplet to the ribosome, and you'll get the protein being built up. So DNA codes the protein, RNA brings it to the ribosome, that's mRNA, tRNA brings the amino acids and joins them together to form the protein. I hope that answers your question. You really do not need to know as much detail as they make it out to be. What you need to know is you need to know DNA replication, but not so much about protein synthesis until A2. I'm sure the notes are fine. Stop worrying. All right. Immune response. Okay. Actually, one sec. I'm going to. Sorry. I've been keeping all the notes I've been making over the last live streams, and I'll see if I've still got the one from earlier. Because if so, I can show you. Because I think that was a reasonably good diagram. Is that it? Sorry, this. I will find it because I am lazy and it took forever to write out last time. But I'm more than happy to go through it. I really hope it was on the first live stream, so it's going to take forever to find. But talk among yourselves, class. Aha, here we go. Oh, God. That's nearly chucked papers all over myself. Right. Sorry about that. Right, here we go. Right, so have a pathogen. It'll either be ingested by a phagocyte, and this will present the antibodies which are on the pathogen on the cell surface membrane. It could infect a cell, and then it will present its antibodies on the cell surface membrane, or it could just be free floating, just expressing its antibodies naturally. Now, it can No, Aubrey, it's fine, fine. That's what I'm here for. I'm here to go over things. Right. So you have antigen-presenting cells, which are phagocytes. You have cells which have been infected, and just the pathogen moving around by itself. Now, it can be detected by two types of cells, T helper cells and B cells. B cells will have complementary antibodies, which will match with the antigens. And T helper cells have complementary receptors, which bind to the antigens. This stage called clonal selection, they find out which ones are the correct ones. Once you've done that, so you have clonal selection, because you're choosing which ones. Then you get clonal expansion, where they replicate. So T helper cells, when the right one's been chosen, will replicate to more T helper cells, which can then activate more B cells and T killer cells. The B cells will split into B memory cells and just normal B cells. Now the T killer cells will just kill the infected cells. T helper cells will then become memory cells, which means if it gets infected again, these cells will automatically clonally expand a lot quicker. You don't need to go through clonal selection, won't take as long. B cells, you get the B memory cells, same function, and then you get the B cells which will release antibodies, which will then also attack the antigens and kill them. Can we go through a lymphocyte diagram, which is in the OCR textbook about this? If by well, that's basically the diagram I've just gone through, Brian. Right. In terms of wording your answers, the way you can do that, practice, and you see how to word your answers. Right. Describe the effects of smoking on page 178. Oh God. Right. With with reference symptoms of. Right. Okay. So the main things you have in cigarette smoke. So just before I do this, let me just check the messages. Okay. Yes, that's what you do. So, main things we have tar, carbon monoxide, nicotine. Right. So, tar. Now, oh God, you asked a reasonably long question here, but right. Now, tar settles in the airways and the alveoli, in your lungs, your bronchs, your trachea, all that, settle in that and the alveoli. Now, the presence of many chemicals in the tar lining on the surface membrane may cause an allergic reaction. That's just a general problem. This will cause smooth muscles in the walls of the airways to contract. The lumen of the airway gets smaller, and this restricts the flow of air to the alveoli. So basically, you get a constriction. But the main problem is the tar will paralyze or destroy the cilia. This means they're unable to move the layer of mucus away up to the back of the mouth. But the tar also stimulates cells to produce more goblet cells in particular, to produce this causes it causes goblet cells to produce mucus, so you've got more mucus being produced and no cilia to waft them away. So you get a build-up of mucus in your lungs. Okay, yes, Brian, these all happen at once. Yeah. Right. So what happens then? You get bacteria, viruses, pathogens of bad stuff getting stuck in the tar, but it's not being removed, which means that the lungs are now susceptible to infection. But also you get the long-term conditions, which is you start getting smoker's cough, which is you're trying to shift all this stuff, so you cough. Doing this will damage the, I believe, smooth muscle. Right, let me just check. Well, it damages the airways, and it's replaced by scar tissue, which is less flexible. The smooth muscle in the wall of the bronchus thickens. This reduces the lumen of the airway, and the flow of the air is permanently restricted. It's a layer of epithelium which is particularly damaged, which then attracts white blood cells because you've got the bacteria and the viruses, which will secrete when enzymes to break through from the blood into the airways. So what do you mean no? Anyway, the white blood cells break into the airways, secreting enzymes and digesting part of the lining of the lungs. This damages the elastic tissue in the lungs, which means that if you lose elastic tissue, it doesn't, spring back, so you've got that problem. King Genetics, anything. Pretty much anything can be asked. I can't see Martin much on the food and health section though. It's probably going to be for the biodiversity one. Anyway, as I was saying, stop distracting me from the current question. So yeah, so we get basically build up of mucus which cannot be wafted because the cilia have been destroyed. So you get bacteria, you get damage, you get smoker's cough, which will damage the lining of the airway. So you get scar tissue, more smooth muscle reduces the size of the lumen. You get pathogens staying in there. So you get you will have white blood cells come and release enzymes, digest part of the lining so they can break through to kill the pathogens, which will then destroy the elastic tissue, which means that you can't push air out as easily. That's a problem. Also, it can cause lung cancer. Isn't good. Now, bronchitis is inflammation of the airways. So this is damage of cilia, overproduction of mucus. So that's that kind of part of the tar problem. Emphysema is loss of elastic tissue. So that's where that comes from to do with tar. The other thing you have nicotine and carbon monoxide. So carbon monoxide, it basically permanently binds to hemoglobin, forming carboxyhemoglobin, which means you've got less capacity for carrying oxygen, which is bad. Yeah, I doubt the immune response could come up. I think the biodiversity module is the one they would ask again from this unit. So anyway, carbon monoxide permanently binds to hemoglobin, so you reduce your oxygen carrying capacity. Nicotine obviously gets you addicted to it. Causes the release of the hormone adrenaline, increases heart rate and breathing rate, causing constriction of the arterioles, raises blood pressure. Constriction of the arterioles, leading to extremities of the body, reducing blood flow and oxygen delivery to the extremities. In extreme cases, it can lead to the need for amputation. It also makes platelets sticky. This increases the risk of a blood clot or a thrombus forming. And nicotine can also mimic the action of a transmitter substance at the synapses between nerves. This makes the nervous system more sensitive, making the smoker feel more alert. How does it cause? It contains. It's not so much to do with the tar, more the fact it contains carcinogenic substances which can cause mutations in the genetic material. The main one is benzopyrene. It's found in the tar, but it's a carcinogenic compound. It enters the lung tissue and can cause a mutation, which will then lead to cancer. All right, I need to go back a bit to. Aubrey, again, can you have to be more specific in your question? Go over classification, speciation, and evolution. That's a big topic in the book to go over. So I need like specific questions from each module, please. Induced fit and enzyme substrate. Oh, good. Finally, something from that module. I like that module. I have to admit, I hate this unit personally. It's not a good unit. The first bit, the biological model. Oh, someone asked me if you need to know the practical biochemistry. Yes, all those biochemical tests you do need to know and could be questioned on. Right, induced fit. Right. So you want induced fit and enzyme substrate complex. Wait, what? There isn't a difference between induced fit and enzyme substrate complex. An enzyme substrate, you have substrate here and the enzyme. All right. Basically, substrate fits an enzyme, forms an enzyme substrate complex. Induced fit just says that, as you can see, it's not absolutely perfect, but when it does go in, the enzyme will change shape so that it fits very tightly around the substrate so it can't just leave. That's what the induced fit hypothesis says. So basically it fits in and is a very close fit. Um, I, I don't know what you mean by happen at the same time. They'll all be going on in your body at the same time. So yeah, I'm reading it for the textbook. Um, right. Right. I need to say this now because I'm getting so many questions. I am a student. I am just like you. I have no. If you can't get the June 2013 papers, neither can I. I don't have them, and I never will be able to get them. In situ, right? I'll answer that in a minute. One sec, because I need to. Um, Actually, that's the next one. I hope that answers your question about induced fit and yeah. Right. Right. So in situ and ex situ. In situ means in there. I have actually done this a few times, but don't worry. In situ means inside their own environment. Um, yeah, I don't know that. Ex situ means you take them out of their environment. So in situ stuff like conservation parks, legislation, making sure that people can't poach them or hunt them. You have national parks. You educate people so they don't kill animals. Um, Repopulation, so you can bring animals back into the wild. Stopping trade. Well, I don't know. I don't know how you're writing your notes. I assume they'll be pretty good. Why send you? Yes, that's what you do with your notes. I've said a number of times, and I've just answered your question, Brian. I've answered it a number of times. Right, back to in situ ex situ conservation. Right. So in situ, yeah. So basically, it's making sure they're staying in their environment. Conservation parks stop poaching and hunting. Making sure they've got a nice environment. There's no pollution. Things like repopulation, teaching other people. Ex situ is things like zoos. So you get to educate people in zoos. You can, like breeding programs. Um, oh, don't worry. People can ask the same question. Don't worry. Um, okay. Ex situ. So that's like zoos. Um, Freezing sperm and preserving it for the future, repopulating programs. You also things like seed banks where you can like store seeds, cool them down for hundreds of years if you can, and then test them, see how they will germinate, and then use them when we need them. No, I do. I'm a right. At least I'm a student. I do not have access to anything. Right. Does that answer your question about ex situ and in situ by both of you who asked? Um, if not, would you like to go over it a little bit more? Because it's getting a little bit manic here. How do you make a hormone? That's an interesting question. How do you make a hormone? Yeah, it's a protein, so by protein synthesis. Lol, people are getting angry. Oh, don't worry. But yeah, are the two people who asked for the in situ ex situ they happy? Okay, good. Cleared it up. Wonderful. Um, right. Um, on a second. You just don't pay. What? It's basically ex situ and in situ. In situ, just think of anything, any techniques which are done for animals in their natural habitat, like preventing poaching and hunting and educating people there, and you know make sure they don't deforest and there's no pollution. Ex situ is anything to do with zoos, anything that takes them out of their environment, to zoos, breeding programs, seed banks for plants, storing them for many generations, things like that. Can I've told you before how to answer suggest questions. I've done it on another live stream. Right. Coenzymes and right, inorganic, right. These are all types of let me just check. Wait. Um, where is it? Right. Yeah, they're all type of cofactor. These are non-protein groups which will, or any substance that must be present to ensure an enzyme control reaction takes place. Prosthetic groups are non-protein bits which permanently bind to a molecule. So a heme group is a prosthetic group. It's iron which permanently binds to hemoglobin, and that's what actually takes in the oxygen. Coenzymes are bits that basically are not permanent. They temporarily adjust to something. Oh, that's a terrible joke. I've heard that joke before, and it's awful. Um, but yeah, so coenzymes, they bind to it for a while, which will change the shape of the molecule, causing it to be able to work. And then once it's done, once it's reacted, it will then unbind to it. And inorganic ion cofactors, these will increase the reaction rate. So in some certain ions will increase the rate. So they'll combine either with the enzyme or the substrate, which makes the enzyme substrate complex easier to form, because it affects the distribution and in some cases the shape of the enzyme substrate complex. So prosthetic groups permanently bind to have an effect. Coenzymes temporarily bind. Inorganic ion cofactors also temporarily bind, but they're ions. Okay, Brian, you don't need to repeat the same question twice. I will answer all the questions. I'm going back. Right. Um, can you talk about Simpson's index and what is the difference between classification and taxonomy? Well, can you talk about the Simpson's index? Can you be a little bit more specific on actual questions? Yes, I can talk about it, but I don't know what you mean by just talk. Yeah, what do you mean by talk about it? But I can do classification taxonomy. Well, they're not really different. Right, no, they're not. They're by definition. I will read a perfect definition, because definitions are always good. Classification is the organization of living organisms into groups according to their shared characteristics. Taxonomy is the study of the principles behind classification. So again, there's not really a difference between them. They're not like completely different things. As in Gymland, they are in cofactors. Look up cofactors. What was I saying? Yeah, taxonomy is basically the study of principles behind classification. Classification is actually sorting things into groups itself. Yeah, that's the definition. King Genetics, if you think it's easy, do you want to answer it? Makes my life a lot easier. Are my playlists up to date? AS, the AS Unit 1 and Unit 2 Biology are. AS Chemistry Unit 2 is up to date. A2 Biology Unit 4 is up to date, and Unit 5 is up to date apart from the 3 videos I haven't done yet. Unit 5 chemistry is up to date, apart from 1 video I haven't done yet. And Unit 4 chemistry it's only got the first 2 videos in it because I'm doing the videos in completely random order and whichever I feel is most important, so that's not up to date. But all the other ones are up to date. All the AS ones are done. I can go over Darwin theory. Yep. Anyway, let me go. Right, where am I? There you go. King Genetics will answer. That's useful. Can you go through colorimetry? Right. Yes. Basically, do you actually want colorimetry? Right. So, what you do is you have an unknown concentration of a sugar. Let's say glucose, because we use glucose. What you do is you want to do the Benedict's test, the reducing sugar test, on known samples of glucose. So let's say, I'll do it here. You might want to do it on 0 mole per dm cubed, 1, 2, 3, 4, 5, whatever, like that. Right. Don't worry about the curve right yet, but so colorimetry is basically you shine a light through your sample of glucose. A photoelectric cell picks up light on the other side that's passed through the sample and will give you a reading showing how much light has passed through it. And you place this solution in the sample chamber between the light and the photoelectric cell in a small clear plastic container called a cuvette or in a special test tube. The more copper sulfate that is used up in the Benedict's test, so basically the more reducing sugar it is, the further the Benedict's solution test goes, which means basically means that the more light will get through. So as the concentration increases, the reaction will go further, so more light will get through, so percentage transmission will increase. And this is called a calibration curve, so we've done it on known concentrations. Then you're given a concentration, let's say this much of it is transmitted, then go down. And so basically you look at your percentage transmission because you do it on an unknown sample, then do a line across to your calibration curve, do one down, it's about 2.7 mole per dm cubed. That's how colorimetry works. Okay. I will answer all your questions, Brian. I just wait. You have to wait your turn because I'm doing them in order I get them. Can I go over water? Again? Moj? Sorry, I don't know how to say that. Sorry, my pronunciation's awful. Can you go over water? What do you want to know about water specifically, like how it bonds together or the properties of water or what? Can I go over Darwin's theory? Yes, I can. Oh God, I'm not a good cameraman. Sorry. Right. Darwin's theory. Right. Well, again, I need to clarify. Darwin said, do you want the actual theory of natural selection, or do you want like his observations and conclusions, or both? Okay, now how do you calculate? Well, it's pretty simple. It's an equation, and you're given it. I don't. Where is it? Right. Right. Oh. Right. Properties of water. Okay. Both. Okay. I'll go through both. I'm using the textbook. Brian, you've asked the same question 4 times. Stop. What? How many times are you asking the question? All right. So basically, diversity equals 1 minus n, which is the number of individuals of a particular species. N is the total number of all individuals of species. You square it and then add them all together. So if I have, I'm going to do it with 2 species. So let's say we have 15 of one species, 25 of another. It becomes 15 over 40 squared plus 25 over 40 squared, which equals, sorry, can't do that in my head. (0.53). 1 minus 0.53 equals 0.47. So that's the diversity. Simpson's diversity is 0.47 for just if you have one species that has 15 and one species has 25. Just an example. All right. Oh my god. I'm using currently the textbook, the main textbook. Right. Right, can you go over water next? I said water. Right, properties of water. Right. Now, as we know, water is polar. Meaning it can form hydrogen bonds. Hydrogen bond forms between slightly positive hydrogen, slightly negative oxygen. Now, the properties of water are extensive and annoying to learn, but you have to. Right. So, it can be a solvent. Now, why this is because since it is polar, anything that is polar or charged can dissolve in it. So that's good. Yes, it's the F212. Right. So solvent, because it's polar, anything that's charged or polar itself can dissolve in it. So sodium chloride, for example, or sodium chloride, Cl minus, which means it can form a bond with the slightly positive hydrogen. Liquid. Water is a liquid, which means it can be materials can move materials around organisms. So like in the blood, liquid in there. Cohesion. Water molecules can stick together, often creating surface tension at the water surface. It also makes long, thin columns very strong and difficult to break of water. So, as we said in the xylem, water cohesion can join it, making it stick in one long column. It also means that small organisms can walk on water. So, like pond skaters, because of tension, they can walk across it. Then you have freezing. When water freezes, it forms ice. Now, the reason why it does this is because, when water freezes, it forms ice. We know that. But when it forms ice, the hydrogen bonds are still there, and you kind of have spaces between the water. So I'm going to say water, water, water. So in a solid, you have these kind of gaps between them, which means that ice is less dense than water itself, which means ice floats. So you have water, ice will be on the top. Now, in cold weather, ice provides a insulating layer. That means the water below isn't going to freeze, so it can still support life. Thermal stability. Well, I believe it's called that they have a very high specific heat capacity and latent heat of vaporization. Those are the two you need to know. So high specific heat capacity and a high latent heat of evaporation. Now these two mean a high latent heat of evaporation means that when it does evaporate, water needs a lot of energy to evaporate. So when it does evaporate and the bonds break, it takes a lot of energy with it. So when you sweat and water leaves the skin, it's taking a lot of heat energy with it. That's important. And high specific heat capacity means it takes a lot of energy to increase 1 gram of water by 1 degree Celsius, which means that it's it's thermally stable. So if you require a lot of energy to actually heat up water. So in the body where you need a constant temperature, so in your body when you need a stable temperature, it can stay at that stable temperature. It won't just suddenly heat up or down at any. It needs a lot of energy to heat up. And water takes place as a reactant to some metabolic processes such as hydrolysis of carbohydrates and proteins. I hope that answers your question. How long have I got? 40 minutes. You wanted Darwin, right? Good old evolution. Right. Darwin's observations were: offspring generally appear similar to their parents, despite, you look at your parents, people can say, oh yeah, I see this, link between you and your parents. We aren't born looking like dogs, and our parents are humans. We all look similar. No two individuals, however, are identical. Even genetically identical twins have differences between them. Organisms that have ability to produce large numbers of offspring, so frogs will produce hundreds of babies, but populations in nature tend to remain fairly stable in size. You don't see frogs spiraling out of control, unless they're released somewhere like Australia. Now, this is the conclusions Darwin made: was there's a struggle to survive. Better adapted individuals survive and pass on their characteristics, and over time a number of changes may give rise to new species, and that since parents produce more children than the population size, yet size doesn't increase, organisms must die. Right, so that's Darwin's theory. Right, then natural selection basically is very easy to get marks on this sort of question. Right, organisms will randomly mutate. So in the genetic code, when it replicates, you'll get random mutations. Ones that are beneficial to the organism are called adaptations. These adaptations will cause organisms to survive over other organisms. So let's say, organisms living in the cold, and they develop, let's say it's a plant living in the cold, and it develops by random mutation a gene for cold resistance, it's going to survive. When other organisms that haven't got this gene will die. That means the cold resistant gene is passed on to the next generation. So that means the next generation has that adaptation, and the ones that don't have it will die. And that's how natural selection works. It chooses the ones with the best adaptations. So I hope that answers your question. Right. I see you missed that page 144 to 145. Oh, don't worry, you don't need to know about that. You really don't. Is that Daniel? No, you don't need to know about it. It's basically just saying enzymes are involved in reactions. Yeah. There's a reason your teacher left that one out. You don't need to know it. That's why. Right, can I go over taxonomy? I believe is the next question. Taxonomy is just the study of the principles behind classification. That's all it is. So learn that definition for one. Right. So this means the study of differences between species. So basically, taxonomy is just looking at how different species, the principle behind classification, so the differences between species. So whether that be genetic or morphological, so appearances, physiological, biochemical, in other words, embryological, so how they are developed, things like that. So it's just, yeah, you don't need to know huge amounts on taxonomy. Aubrey Reed, a selection pressure. That is like when I said, cold resistant. A selection pressure would be the cold, the temperature. It is something which forces an adaptation to be needed, basically. So these plants will need to have cold resistance, or they will die because it's too cold. So the ones that have that can respond to that selection pressure with an adaptation are the ones that survive. Yeah, you don't need to know too much about. Basically, know the definition for taxonomy and notice like yeah. So taxonomy is just basically looking at the basically the differences between species. Phylogeny is all to do with evolution, and looking at evolutionary relationships between species. So how closely related is one species to another, and this is when tree of life. So for example, these two species are much more closely related than those two species because they're closer on the evolutionary tree. Yeah, you really do not need to know too much about it. You know the definitions and basically kind of be able to explain it. Mohammed, I have gone over the immune response, yeah. One second. Right. Sorry about that one. Right. So I'm having to read through the backlog of all the questions. Right. Brian, I've already done that earlier, talking about seed banks. Right. Vaccinations and passive/active immunity, not right. I really this some of the stuff in this module is so boring. Yeah. Anyone agree with me? Anyone else think this is a completely boring module, particularly the food and health bit? Right. So, before vaccination, I'm actually going to active, passive, natural, and artificial. Now, active immunity is achieved by the activation of the immune system. So you have to actually cause your immune system to go into response. So basically, you need a disease. Passive immunity is when you're kind of given antibodies. You're given things which will, give you the immunity straight away. Natural immunity is gained just naturally, and artificial is artificial. Makes sense. So we're going to look. I'm going to do active immunity for both natural and artificial. Natural active immunity is when you basically get the disease. You get the disease and you produce antibodies and your immune system works. Artificially active immunity is basically when you're vaccinated. When you're vaccinated and you are so your body kind of has that response, but not properly. It's not naturally induced. But yeah. So natural actives when you basically get a disease. Artificial actives when you get a vaccination. Natural passive is when when a baby suckles its mother, it gets those antibodies directly from the mother, and you kind of get protection from the mother. Artificially passive immunity is when you're injected with antibodies. So passive immunity is when you're given antibodies. Natural from your mother, just born as a baby. Artificial when as an adult you're injected with the antibodies. Active immunity is when you have the disease. Natural is when you actually get the disease, and artificial is when you're given a vaccination. So what is a vaccination? Before I answer that, let me just check the chat. Yes, 214 is the best. Brian, as I said, I've already kind of gone through that. Yeah, Daniel, I'm right. Thanks for that, Brian. If you actually noticed, I did answer your question earlier. There is no need to be rude. Because if you are rude, I can just ban you. So please do not be rude. Well, anyway, vaccinations. Basically, vaccination is deliberate exposure to antigenic material, i.e., you're just given a small version of the disease, or you're given the antigens. So the antigens which will then cause the response. So you can be given the antigens, then the same response will happen because they'll bond to lymphocytes and will then trigger that response. So you'll get the B cells being made, the T cells, the memory cells being made. And you're blocked. There you go. I'm sorry about that. Okay, genuine question. If any of you do not like the way I'm doing this, feel free to say. But I aim to kind of use the book to explain it, but then I explain it further in my own words and help. Oh well. But yeah, are you all finding this useful, the way I'm doing it? Okay, great, thanks. A bit more detail, Alice. Ooh, criticism. Um, okay, great, thanks. F213 does exist. It's the practical unit, the AS practical unit. Right, where was I? So I've completely lost track of what I was doing or where I am at all. So I'm going to say, any questions you previously asked, can you just ask them again if I haven't answered them yet, and I will now answer them. So yeah, sorry if you have answered the question already. Please ask it again if I haven't answered it, and I will answer it now. I've kind of lost track. Uh, as that is, yeah, as you said, that is a lot to do. I mean, um, is that, and you do not understand anything to do with them, any of them at all? If you do understand, can you ask like specific questions from them? If you genuinely do not understand all of them, A, have you watched my videos? And B, if you have and still don't get it, can you email me and I will go through it on over my email, just because that will take the rest of the time to explain all four of those sections. So yeah, do you understand? Is there certain bits you don't understand? Um, if you don't understand all of it, have you watched my videos? And if you have and still don't understand it, email me. Thanks. What is the practical module? Have you done practical exams? So you have a quantitative, a qualitative, and evaluative. It might be called ISA. It's that. It's basically you have to do those practical exams. I haven't gone over inhibitors. I'm more than happy to go over inhibitors. But do we need to know about finding new drugs? No. No, I don't think so. Right, King Genetics. I'm actually going to say pandas are one of my all-time favorite animals in terms of the animals, but yes, they should have gone extinct. They are evolutionarily a failure. I mean, they are carnivores who only eat one sort of food, which is a plant. They don't. They're not very sociable animals. They are very specifically, adapted to one thing and one thing only. They can only reproduce 3 times a year. Even then, they don't want to. And if they do even reproduce, it's usually very unsuccessful. Um, and I know lipids are. Okay, yeah, I'll do yeah, that. I can't say that word, so don't expect me to. Right, so I'm going to do inhibitors first, because that was asked. Right, 2 types of inhibitors: competitive inhibitors, non-competitive inhibitors. So, substrate enzyme. Competitive inhibitor. A competitive inhibitor is one that has a similar shape to that of the substrate, so combined complementarily to the enzyme. So in this case, it'll be like that. Prevents blockage, or blocks the active site, so the substrate can no longer bind to it. And that's competitive. Non-competitive is one that binds away from the active site but changes the shape of it. So in this case, you might get which means no longer can the substrate bind to the active site because the shape's change, so it's no longer complementary. So competitive binds to the active site, blocks it, and so the substrate can no longer bind. Non-competitive binds away from the active site, changing the shape of the active site so the substrate can no longer bond to it. I did cofactors and coenzymes a bit earlier. It is a whole unit in AS, yeah. Unit 3. Um, my favourite animal is the African wild hunting dog. I love them. Sorry, random thing. Does it answer the person's question about inhibitors? Whoever asked about inhibitors? Mohammed, does that answer your question, or did you want to know something else about inhibitors? This is just about how inhibitors function. Right, thanks, good. Right. Atheroscler. I hate that word. Yeah. I'm sorry, you probably all get very angry at the fact I cannot say it. Right, where is it? Where is it? Right. No. Right. basically you can get buildup of fats, cholesterols, which are caused by LDLs, which are low density lipoproteins. The low density lipoproteins will deposit the cholesterol in the lining of the lumen, not the lining, the what's it called? Inner lining. Oh, inner lining of the artery. That's it, artery. That's the word I was looking for. So inner lining of the artery, the endothelium, and it does this because carbon monoxide can damage the inner lining of the arteries. Same with if you have high blood pressure and cause damage, which means that the damage is repaired by white blood cells. But this encourages the growth of smooth muscle. But while you've got that damage, you can get low density lipoproteins, so cholesterol and fatty substances building up. So you kind of have damage. The fats and low density lipoproteins and cholesterol can get in it, be deposited in it. Smooth muscle can grow as well, and this basically creates this shape. You could have something like, so that's the artery, fatty buildup, which reduces the size of the lumen, so it kind of blocks it, and this is called a thrombus or a blood clot. And actually, you didn't ask, you just asked about the ather thing. But the buildup of atheromas occurs under the endothelium. It can break through the inner lining of the artery and eventually forms plaque, which sticks out in the lumen of the artery. This leaves the artery wall rougher and less flexible than in the healthy artery. So basically just carbon monoxide damages it, so does high blood pressure. As it's being repaired, you encourage the growth of smooth muscle and the deposition of fatty substances, cholesterol, low density lipoproteins, which causes a kind of prominence to be produced in the lumen, reducing the volume. And yes, the end result is coronary heart disease. Only if it occurs, actually, no, not necessarily. Only if it occurs in the coronary arteries. If it doesn't occur in the coronary arteries, so it occurs anywhere else in the body, you will not have coronary heart disease. What is my favorite animals? I'm a very big dog person. I love the way they work. I think they look beautiful. They're beautiful animals, and I like the way they hunt together. I just think they're pretty cool animals. Right. Does that answer everyone's question on the long, complicated word I cannot say? You need to know in about as much detail as I said. I think to be able to describe it, probably for 3 or 4 mark questions. So you can make 3 or 4 good points about it, then you'll be fine. Can you explain the hierarchical system, Animalia, the genus, and how to? Do you mean the order, kingdom, phylum, class, family? Kingdom, phylum, class, order, family, genus, species. Do you mean that? Per, can you kind of give me an example of what sort of question you mean? Really? They do that? Wow. What is meant by genus? I don't know how to define it. Oh, I hate words like this where you know the exact meaning but you have no idea how to define it. Does it have an? Okay, genus. It has a definition. Taxonomical group used in the classification of living organisms. Species that are similar are placed in the same genus. Okay, so it's a group of similar species, but not the same species. It's basically right. Our genus name is Homo, meaning man, not Homo, which means same. People call us Homo sapiens, but actually Homo sapiens, because Homo means man. So our genus is Homo, and our name is species name is sapiens. Underline it because it's a Latin word. Now, in the past, there have been other Homo species. There's Homo habilis, Homo erectus, Homo ergaster. I believe Homo neanderthal was one. I think Neanderthal was in our same genus. I believe so. Homo heidelbergensis. All these different homos. And they all share characteristics. They weren't the same species. They couldn't reproduce to produce fertile offspring, which is a definition of species, except maybe you get some hybrids of Neanderthals and humans. But genus is the Homo name. So our genus is the Homo name, is a group of similar species. Okay, but call me Jay. I did that literally right at the beginning of the live stream. I went through that right at the beginning of the live stream. Pair, yes. Is that okay? I assume that means right. Well, it's the order is kingdom, phylum, class, order, family, genus, species. Just need to remember that. Generally, they'll give you quite well. You just need to literally learn that. So I don't really know how to help with that. Just recite it yourself till you can go kingdom, phylum, class, order, family, genus, species, and then yeah. Unless can you find an exact question like that, and I'll go through it. Levels of protein structure, right? Primary structure, secondary structure, tertiary structure, and possible quaternary structure. Right. So you have proteins. Structure of protein. That's the general structure of amino acid. You're welcome. Yeah, there are loads of ways to remember kingdom, phylum. I literally just remember it the way I've, yeah, that. Now, the primary structure of protein is the specific sequence of amino acids. So I'm going to just say you could have A, B, D, A, F, C, 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 and it ends there. That could be the sequence of amino acids. Now, that's coded for by the DNA code and sequence. Okay, I will keep going. Do you want to know about condensation reactions and how proteins bond together? Well, I'll continue. I'll go back to it at the end. Now, the secondary structure is the coiling and plating of parts of the polypeptide molecule. So you could get two sorts. You can get alpha helixes, which is basically when you get this. So you could have A to B to D. Quite hard to draw it like this. A to F to C, C to C to end. So you can see you kind of got that alpha helix. And that's caused by hydrogen bonds forming between R groups that contain H, OH, and R groups that contain O. You get hydrogen bonds, as we discussed with water. King Genetics, you very clearly do A2 chemistry, but for A-level biology, we do not need to know about zwitterions. But yeah, don't confuse anyone who's not doing A2 chemistry. But anyway, so that's alpha helixing, or you can get beta-plated sheets, which I don't know what they look like, but it's another form. I'm guessing it's probably something that looks a bit kind of like that, with hydrogen bonds forming between them. That's secondary structure. I'll leave that there in case you want it. Tertiary structure is when you get different R groups acting together. So R groups that contain plus and minus charges form ionic bonds. R groups that contain this can form hydrogen bonds. So I said about OH and then the O. R groups containing sulfur can form disulfide bridges, and then other ones which are hydrophobic or hydrophilic. If they're hydrophilic, they'll go to the outside of the molecule. Hydrophobic will go to the inside of the molecule. And basically, the tertiary structure is the 3D shape you get of the polypeptide. Okay, Per, I'll go answer your question a bit. Okay, good, you know about bonding. Right. So you've got the tertiary structure, which is caused by all these different bonds and the other hydrophobic, hydrophilic interactions, which causes the 3D shape to change. Quaternary structure is literally when you get more than one polypeptide chain acting together. So let's say overall we've got a protein which looks like 3D structure looks like that. It might also form bonds. That's a really bad bond, but it might form bonds with this chain. So now I've got 4 chains. That's the quaternary structure of the protein. So primary is the sequence. Secondary is the coiling or beta plating sheets using hydrogen bonds. Tertiary is between ionic hydrogen disulfide and hydrophobic hydrophilic interactions, creating the 3D structure. And quaternary is when you have more than one chain connected together. No, God, no, not skeletal formula. God, no, we're not doing skeletal. Right. Answer. Can we go through collagen, hemoglobin? I've done that in 2 videos now, so no, I'm not going to go through that. But also because that came up in the last paper, the paper I did, the June 2013 paper. So I highly doubt it will come up again. But I'm more than happy to go through between starch, cellulose, and glycogen. I'm more than happy to. Right. Firstly, before we go through, does that answer whoever just asked the question about protein structure? Yeah, I thought mono. Yeah, I was right. Mono is just the old name for prokaryotes, isn't it? Correct me if I'm wrong there. I want to say where am I? Okay. Right. Starch. Where am I? Right. Well, these are all made up of starch and glycogen. Are all made up of glucose molecules. Starch is in plants, glycogen is in animals. So this is when you have amylose and amylopectin. Amylose is just a chain of alpha glucose, I'm just do A's bonded together. That's amylose. Amylopectin, if I'm not much mistaken, is very similar but has branches on it. Let me just check. Yeah, can someone confirm that, please? Yeah, I'm pretty sure. Isn't amylopectin when you have 1,6 glycosidic bonds? Can someone confirm that for me, please? But yeah, so basically, amylose, I'm pretty sure, because this is 1,4 glycosidic bonds. I'm pretty sure amylopectin has 1,6 as well. So it'll be look something like this, a bit like that. Wait till someone confirms that. Please, someone confirm that. Yes, I got a yep. Right, great, thanks. Just making sure. Right, starch consists of a mixture of amylose and amylopectin, and that's basically the structure. Glycogen because, right, let's say we have starch, which looks like this. Amylose will have shorter 1,4 glycosidic bonds. I'm sorry, not amylose. Glycogen has shorter 1,4 glycosidic bonds, and it has more 1,6 ones. So here you might just have one. Here you could have, you know, many more, much more, a lot more. Yeah, I've got many yeps. Good. Yeah, so there's many more branches. So starch, long bonds, few branches. Glycogen, short bonds, more branches. This means that glycogen is more compact than starch, and so glycogen can dissolve. I hope that answers between glycogen and starch. Cellulose is completely different because it is structural. Now, I hope that answers your question. Please say it does. Cellulose is between beta, which means you get a perfectly straight line between beta glucose. And basically you get these long chains which form cross-links with other chains. And you get all these chains which form microfibrils, and then you get loads of these microfibrils joining together to form macrofibrils. So that's essentially what it is. So cellulose is beta glucose joined together. Okay, good. I was correct for amylopectin. Yay. Okay, good. I was completely correct on that. I like it when I am correct. It's occasional and rare, but I like it. Yeah, so cellulose is beta glucose joined together. Those kind of long chains are joined by cross-links to other chains, I believe hydrogen bonds, yeah. And then those chains are joined together, you get several hundred, you get microfibrils, you get loads of them joined together, you get macrofibrils. That's cellulose. Does that answer your question? And finally, I will end with the June 2012 question 2A. June 2012, 2A. And no more questions, please, because I'm running out of time. 2A. Right. Oh, this is classification. Right. Wait, what? Okay. Kingdom. I don't think I have actually. I don't think I've done LDLs and HDLs. No. Call me Jay. Email me on my email. I'll go through them with you. But I'm just going to go through final question 2A. Right. It says here kingdom prokaryote blank fungi plant blank animal blank. Now you know that means you need to know the 5 kingdoms. So you know that Protoctis has been left out, and you know that plant has been left out. So one of those is plant. One of them is Protoctista. Now, let's look at the first blank kingdom. We know membrane organelle present, cell wall sometimes present, composition varies, and it's heterotrophic and autotrophic. Now we know plants are always autotrophic and they always have cellulose cell walls. So that must be Protoctist, And the second blank kingdom must be plants. We know the cell wall must be cellulose for plants, so we got that one. First row, types of nutrition for prokaryote can be heterotrophic and autotrophic. Third row, fungi, membrane-bound organelles are present. And finally, animal membrane-bound organelles are present. So the things you need to know is the 5 kingdoms, whether each kingdom is autotrophic or heterotrophic or saprotrophic even. What the cell wall is made out of, they love that one. Stuff to do with organelles, does it have organelles, and any other very particular defining features, like how they reproduce or if they can move, things like that. I hope that answers your question. Sorry, Alice in Gymna. Amylopectin makes up starch. You have amylose and amylopectin in starch and glycogen. They're basically the same thing. But in starch, you have longer bonds and you have less amylopectin branches. That's the difference. I hate that too. It drives me mad. Biology is a science. I mean, by definition, it is a science. And yeah, I don't know why. So chemistry teachers want to feel better about themselves. The same reason why biologists say psychology isn't science. Because it isn't. What? No, biology is a very clear found science. And without biology as a science or a subject, we would not be anywhere today. I mean, there's so much we couldn't do without biology. I mean, there'd be no medicine, the world would have died. There'd been so much we couldn't do. I mean, chemistry, it's very useful, but chemistry becomes pointless unless you actually have biology to use it for. Without the knowledge of biology, all the chemistry we can do becomes pointless. We just, you end up creating loads of toxic chemicals that kill everyone. But did that answer your question? I don't know who asked the. Pear, did that answer your question slightly? It's basically the thing you need to know about classification is page 202. Everything on there. Page 202 is what you need to know about the 5 kingdoms. But I hope yeah, well, we all know that biology is not all chemistry. I'd like to see a chemist who can, well, I'd like to see how a chemist could justify stuff like classification, evolution, biodiversity, count that as chemistry. I mean, you can go to a chemist and go, it's all physics, so. Yeah, I'll say to you can say to all chemists that chemists just do is blow things up. So yes, I hate it when people do not say biology is a true science when in fact it very much is. And I would like to see what would happen to the world without science, without biology in particular. Without physics, we lose luxuries. Very little in physics do we actually need. 100% need. Like no other animal needs to have all this, electricity. We don't need it. We don't need cars. They're useful, but we don't need them to survive. Chemistry, I would like to see, how do we need chemistry to survive? Well, yes, in terms of drugs, but only when it's linked with biology. Things, like working out the heat in a system, I don't think we need that. That's not needed. It's useful for many things, but we don't need it. Biology, looking at ecosystems, making them survive, finding drugs, seeing how the human body works, we do need that to survive. Without stuff like that, then we would be deeply ruined. Call me Jay, or you're getting blocked if you say that again. But psychology is isn't it? The problem with psychology is that it's kind of a load of theories, none of which have been proven. I mean, they've got 6 theories for how the mind works, and none of them are proven at all. You don't get that, but anyway, I think time to call that to an end now after a very weird and a slightly annoying live stream. due to certain people, but I hope that helped you all. Apart from that one annoying guy, I had a lot of fun, as I always do, and I hope it helped you all and you got a lot from it, because this will be the last AS live stream I'm doing. It'll be the last AS anything I'm doing, to be honest. I'm focusing on A2 now. I've done all the AS videos I will do. All the AS biology videos are done, completed. And all the Unit 2 AS chemistry ones are done as well. So I've done all them. If anyone needs any specific help, either comment in the specific video or email me. And I will help. But in terms of AS live streams, I'm done. And I know your exams are in about a month and a half for Unit 2. So yeah, just email me if you need any help. And I wish you the best of luck. And please, once you've done the exams, do tell me how they went and if any questions came up that threw you or you liked. So yeah. So basically, email me if you have any help. I hope this all helped you now. Good luck for the exams. Work hard, revise hard. Yeah, put in the hours, and you get out what you put in. If you work really hard for the next month and a half, you will get good grades. Not necessarily, you might not get good grades, but you have a really good chance of getting good grades. So try your hardest, work hard, come to me if you need help, and I hope you all do the best. So thank you. Yeah, I doubt I'll be speaking to you again over a live stream unless any of you are in A2. But thank you, and good luck, and goodbye.